Hello everyone and welcome to today's class. We'll start in 5 minutes so that more people can join in. Yeah. guess we can start now. So I'll give a quick recap of what we have studied in the last class, and today in general we'll have a very short class with respect to GPCRs, and we'll see what exactly are GPCRs. So if you remember, we saw the overall view of how proteins are sorted, especially membrane proteins, right? And this was the overall flow of it. For example, these are the proteins which are being translated in the cytosol. If they have a signal sequence, they will be sent along with the RNA to the ER, and then from here they will be translated and folded into their functional 3-dimensional structure. They'll be again sent to Golgi complex, and from there they'll be sent to wherever their destination is. Okay, so this is a very critical process, which is like intracellular trafficking of molecules within the cell, and the proper localization of such proteins is necessary to ensure the proper functioning of the cell. Correct? Then we also saw that a lot of research has been taken place, and a lot of Nobel Prizes were also awarded for actually deciphering the steps which are involved in protein sorting, as well as intracellular transportation or intercellular transportation, right? So how we know that proteins are transported from one site to the another site inside the vesicle? So basically, if there are a lot of work has been done, so if you look at this simplistic overview of it, so if you have a membrane on which the protein is being translated, so if you add a detergent, so what the detergent will do, the detergent will disrupt the vesicle, right? Because at the end of the day, what vesicles are? They are the membrane structure. So if you remember this representation, this one, right? Vesicles are also formed from the membrane itself, so they have all the qualities and the characteristics of the membrane through which they are being generated. Okay, so if you disrupt these vesicles, what will happen? The protein will be exposed to the surrounding, or the buffer in this case, because these experiments were taking place in vitro. And if you then add proteases, what proteases will do? Proteases basically digest the protein, correct? So you will get digested protein. On the other hand, if you don't add any detergent, what will happen is that protein will be safe inside the vesicle, inside the membrane. Okay, so proteases won't be able to enter inside the membrane, and hence you won't get any degraded or digested protein, correct? So this basically is the culmination of years and years of work, and it showed us that proteins are actually transported within the cell inside the vesicles. Okay, you can actually see that. So if this is the photograph of that, so these are the ER lumen, these are the ER membrane, and you can see that these are the ribosomes, right? Attached ribosome, and these are the free ribosome. So on free ribosome also protein is being synthesized, and once it is attached to the ER membrane, it will start secreting the protein or translating the protein into the lumen of the ER, correct? But the question exists is that why is that only non-cytosolic protein should be directed to the ER? Because all translation, if you look at the compartmentalization of functions within the cell, the protein is being the initiation of translation occurs in the cytosol at the end of the day, correct? Then why some of the translating machinery being sent to the ER while the other are not sent to the ER? See, you can see here. So this was the one path, this is another path. So you can clearly see if it doesn't go towards the ER path, they'll translate in the cytosol, the protein will fold into its functional state here itself, then further it will be directed towards wherever it is supposed to go. Okay, so the question exists is that why is it that only non-cytosolic proteins should be directed to the ER? Okay, so a lot of work goes into this, and as you have seen during the earlier discussion of this particular topic, that I was taking these scientists' names, Sabatini and Palade. So earlier showed, both of them actually showed that it is large ribosomal subunit that is instrumental in attaching the ribosome to the ER membrane. Okay. That instrumental in attaching the ribosome to the ER membrane, Palade concluded that there is a vectorial transfer of nascent polypeptide chains from the ribosome to the microsomal membrane. Palade could not molecularly define the interaction between the ER membrane and the ribosomal subunits. Okay, so what happened is, as you have seen that Blobel and Palade, they actually worked together. Palade was a student of Blobel. So Blobel suggested that there is a signal hypothesis is there, that the proteins and the machinery of translation is subjected towards the ER and then the secretory protein pathway occurs. But even till that time it was not clear that what is that which brings some of the protein towards the ER and some of the protein do not get directed towards the ER. So from there the Palade actually started. The free and ER bound ribosomes are identical because at the end of the day the translating machinery is same, correct? So the hypothesis which Palade came out with the work of Blobel and all the other work in the field which was taken place at that time is that what they said is, we postulated that all mRNAs for secretory proteins code for a signatory amino terminal sequence element that is recognized by a soluble factor that in turn binds the nascent chain ribosome complex to the ER membrane. Correct? So just put yourself in their shoes. What you know that there are certain proteins which are directed towards the ER, while there are certain proteins which are not directed towards the ER, but the translating machinery is same, correct? So if you put yourself in their shoes, what you can actually understand is that the machinery is same, the place is same, yet a few moments into the translation some of the proteins are being sent to ER. On the other hand, after some time into the translation, some proteins are not sent into the ER and they stay in the cytosol. Correct? So there has to be something in the translating protein itself which targets the translating machinery towards the ER. I hope you understand, right? So what I'm trying to say, so the hypothesis states that we postulated that all mRNAs for secretory proteins code for a signatory amino terminal sequence element that is recognized by a soluble factor that in turn binds the nascent chain ribosome complex to the ER membrane. Again, I'll try to simplify it for you guys. This is the location of translation. It is same for all the proteins, irrespective of whether they are membrane proteins or they are cytosolic proteins. Correct? Now, the observation was there are certain proteins that few moments into the translation they have been sent towards the ER, while there are some proteins which are not sent towards the ER but they stay in the cytosol. This was the observation. Correct? So, what another observation and the fact which we know is that the translating machinery is same. Correct? So what Palade and Blobel hypothesized is that there has to be something in the translating protein which dictates that the translating machinery either should be sent towards the ER or it should not be sent towards the ER. Right? So now as we understand this hypothesis, we postulated that all mRNAs for secretory protein. Now here secretory proteins means the machinery, the proteins which are being sent towards the ER. Correct? So Palade and Blobel said that we postulated that all mRNAs for secretory proteins code for a signatory amino terminal sequence. Now why we are saying it is amino terminal sequence? Because protein translation takes place from N terminal to C terminal. Correct? So after few minutes or seconds into the translation, the machinery which is being sent towards the ER should have something different towards the end terminal, which is being recognized by something which is present in the cytosol, which is directing this whole machinery towards the ER, right? So again, we'll try to understand this hypothesis. We postulated that all mRNAs for secretory proteins code for a signatory amino terminal sequence element that is recognized by a soluble factor. Means there has to be something, there has to be a soluble cytosolic protein which is recognizing this additional thing in the translating protein, which binds to the nascent chain ribosome complex. Why? Because the whole complex is being sent towards the ER, right? It is not only the protein which is sent towards the ER, it is the whole translating machinery which is being sent towards the ER. Binds the nascent chain ribosome complex to the ER membrane. Correct? So this is the hand drawn representation of how Palade and Blobel actually imagined the transportation will take place. So you can see that this is the subunit pool. Subunit pool means when there is nothing to translate, both the larger subunit and the smaller subunit of ribosome stay separate, right? So this is the subunit pool. The moment it receives an mRNA for translation, the units will combine on this mRNA and they'll start translating. And few moments after the translation, we'll have a signal sequence which will make sure that this whole translating machinery should be directed towards the ER. So this signal molecule or signal sequence will be identified by a binding factor or the soluble factor or the proteins which we know of today, and it will attach this whole translating machinery towards the ER membrane, and then the translation will take place inside the lumen of the ER, and as soon as the translation is over inside the lumen, the subunits will dissociate and the whole process can start again. Okay, these are some of the very simplistic material which you guys can go through to understand more about the intracellular trafficking and how the translation of secretory proteins takes place. Okay, so again emphasizing that how the intracellular transportation takes place and how we know that it is via membrane only. So this is the in vitro way of showing that proteins do get transported from one segment to the other segment via inside the vesicles. Okay, so if you have microsomes are nothing, these are just the membrane bound, these are just membrane structures which have the translating subunit attached to them. So if you add detergent, as detergents are like lipid molecules through which the membrane is being made, the detergents will disrupt the membrane, and then if you add proteases or enzyme which digest protein, what you will get? You will get the digested protein. On the other hand, if you don't add any detergent, meaning the membrane is still intact, right, and then you add proteases, what will happen? The proteases won't have any access to the protein and hence it won't be able to digest. So this actually shows and depicts that the transportation of proteins, especially the ones which are in the ER lumen or the secretory type of proteins, that takes place inside the microsomes, or in the way the it gets blebbed outside the membrane and they'll get merged into the Golgi for their sorting purposes and wherever their target is, the later aspects of the secretory pathway. Okay, so again this is another simplistic way of showing the same thing which we are saying. So the thing which we were talking about, the soluble factor, right, or the binding protein or the binding particle, so now we know it is called the SRP. Okay, signal recognition protein. So this signal recognition protein, it basically recognizes the mRNA and translating protein complex. The translating protein complex which has the signal peptide gets bind to it, pull the whole translating subunit along with the mRNA to the ER membrane, gets attached to the SRP receptor, and lead the translation of this protein inside the lumen of the ER membrane. Okay. So if we look at the basic proteins, how they are being targeted, so most of the things you won't so this is a very simplistic overview of it. We have not discussed much about the SRP, we have not discussed their behavior, that what type of proteins they are, so we should not worry about it. The only thing which you should remember is that the intracellular transportation is by vesicles. Vesicles are nothing, vesicles are just the membranous structure which are generated from the ER, from the Golgi for the transportation purposes. Okay, so we can go through these points. So targeting proteins to and across the ER membrane, so basically it is the protein sorting. Okay, so synthesis of secreted proteins, integral plasma membrane proteins, and proteins destined for the ER, Golgi complex or lysosome begins on cytosolic ribosome, which become attached to the membrane of the ER, forming the rough ER. The ER signal sequence on a nascent secretory protein is located at the N terminus and contains a sequence of hydrophobic amino acids. So why we are certain that they are hydrophobic amino acids? Because if it's a membrane protein, right? Secretory protein, we are talking about membrane proteins here. And that signal needs to get recognized by the SRP. SRP will bind it to the ER membrane. Hence, that signal sequence will eventually needs to get inserted into the membrane. And membrane environment is highly hydrophobic. It won't support any polar amino acids. Hence, the signal peptide will have a hydrophobic amino acid residues. Okay. In co-translational translocation, the signal recognition particle, that is SRP, first recognizes and binds the ER signal sequence on a nascent secretory protein, then is bound in turn by the SRP receptor on the ER membrane, thereby targeting the nascent polypeptide chain ribosome complex to the ER. SRP and SRP receptor then mediate insertion of the nascent secretory protein into the translocon. Hydrolysis of two molecules of GTP, we have not discussed this. Why? Because we have not discussed the GPCRs, which we gonna discuss today. So till here we have discussed, and this is the overall view of how secretory proteins are translocated or how they are sorted. For sorting purpose, they need to first get into the ER, from ER to Golgi, and from Golgi to wherever their destination is. Okay, so this is the overall picture of membrane proteins and their function. Okay, so if you can see, we have discussed different types of transportation, right? So facilitated diffusion is basically diffusion which will happen across the membrane along the concentration gradient, but with the help of protein. Okay, primary active transport, this is the one which will happen against the concentration gradient, and it will require direct association of energy or utilization of energy. Correct? Secondary active transport is, it will get transported by using energy, but that energy won't come from the ATP. It won't be a direct source of energy. Okay. Then we have ion channels. Ion channels basically transport ions across the membrane via along the concentration gradient. We have ionophores also. So ionophores are nothing. They are just vesicle type of transportation, in which there will be a vesicle. Inside the vesicle, the ions will be secured and they will easily diffuse through the membrane, and the ion will be released, and the ionophore entity, which is basically a membrane bound formed structure, it will diffuse out and again it will perform its function. And simple diffusion, obviously it doesn't require any association of protein. Small molecules can easily pass through the membrane. So till now we have discussed one function of the membrane protein, which was transportation. then we have seen how at the end of the day these proteins are getting assimilated into the membrane on the first hand, because the functions are important for the survival of the cell. If cell is not able to transport substrates from inside to outside or outside to inside, the cell won't be able to survive. Correct? So for basic survival, membrane proteins are necessary. But how membrane proteins are generated or how they are assimilated inside the membrane, we were not aware of, and hence we took our understanding from the basic transportation towards how the membrane proteins are sorted or assimilated into the membrane. Correct? Now we will switch gears a bit and we'll see another function of membrane proteins. You remember there were 6 major functions of membrane protein, right? So one of them was transportation. So if you don't mind, I'll quickly go to that slide of the major functions of membrane protein, because it's been a while and we have not discussed it. Yeah, this one. So there are 6 major functions of membrane protein, right? We should keep this in mind always. So the enzymatic activity again will be performed by the proteins which have enzymatic activity and they are membrane bound. Then signal transduction, then transport, then cell-cell recognition, then intracellular joinings and anchorage attachment. So till now we have seen one subset of such major function, which was transport. Correct? Now we are switching gears to another major function and important function which membrane proteins perform is signal transduction. So signal transduction is what? It is basically chemical messengers interact with membrane protein binding site to send forth signals. Okay, so you can see this is a simplistic cartoonic way of representing this. So we gonna now switch our gears, and instead of transportation, which is the major function of membrane proteins, we'll try to learn about another major function of membrane protein, which is signal transduction, because now we know how proteins are synthesized inside the cell, especially the ones which are supposed to be on the membrane, how they are translocated, how they are translated, and how they are assimilated into the membrane. Now they'll perform different functions. First function was transportation, and now the function which we want, which we are focusing on, is the signal transduction. Okay. So all cells have specific and highly sensitive signal transducing mechanisms which have been conserved during evolution. A wide variety of stimuli act through specific protein receptors in the plasma membrane. In all types of signal transduction, receptors bind the signal ligand and initiate a process that amplifies the signal, integrates it with input from other receptors, and transmit the information throughout the cell. If the signal persists, receptor desensitization reduces or ends the response. So we'll break down this statement, because this is basically telling us the whole signal transduction phenomena in 3 lines. Okay. So what it says that in all types of signal transduction, right, there is no exception to this type of signal transduction function of the membrane proteins. Receptors bind the signal ligand and initiate a process that amplifies the signal. So what we mean by saying this is that, for example, you have just eaten, right? So you have a lot of glucose in your body. So glucose sensing proteins which are present on your membrane, what they'll do, they'll sense that a glucose molecule is nearby. That glucose molecule is a signal ligand for this protein. Okay. So that particular ligand or signal ligand will bind to the protein. Now, if we say that if we have 100 molecules of glucose, then all the 100 molecules, if they are sufficient enough in initiating the signal transduction or initiating the response of the cell, so don't you think it will take a lot of time? For example, if there are 20 transporters or 20 receptors on the membrane, but there are 100 molecules of glucose. So it will take a lot of time to take all the glucose molecules one by one via these 20 proteins, and hence chances are the response which we want, it will take a lot of time for our cell to acclimatize. So what happens is, as soon as the signal ligand or a small molecule lands on the receptor, it starts downstream signaling. So the main purpose of downstream signaling is to amplify the signal which we just received. Now this amplification won't necessarily be to increase more glucose molecules, because it is not possible. We are having food so that we can get glucose, correct? If our body is sufficient enough in making its own glucose, then on the first hand we don't need any food to eat. We eat food for energy. Energy is being extracted from the chemical bonds which are present in small molecules like glucose, correct? So receptors bind the signal ligand and initiate a process that amplifies the signal, integrates it with input from other receptors, and transmits the information throughout the cell. If the signal persists, receptor desensitization reduces or ends the response. Okay. Multicellular organisms have 4 general types of signaling mechanisms. Plasma membrane proteins that act through G proteins, receptors with internal enzyme activity such as tyrosine kinase, gated ion channels, and nuclear receptors that bind steroids and alter gene expression. We're gonna focus on G coupled receptors. Okay. Before going through the theory of G protein coupled receptors, I will show you a very brief video of the overview of G protein coupled receptors, so that when we are understanding and learning about G proteins, you guys have some image of G proteins, and it will help you in getting better understanding and learning of G proteins. Okay. So let me, oh, not this one, wait.
आ गया ज्यादा बड़ा डेटॉल, 33% ज्यादा बड़ा डेटॉल, लंबी सुरक्षा.
In a world where academic deadlines meet the blank page.
Grammarly?
Hi, yes. With Grammarly by your side at every step of the writing process,
Yeah,
you can
I
feel
hope you
confident
guys can
submitting a
see
top-notch
my
paper.
screen, my YouTube.
Welcome to Ideation Station.
I'm feeling stuck on this topic for my biology paper, and I...
G-protein coupled receptors, GPCRs, have central roles in many physiological functions and diseases. This diverse superfamily includes over 800 members, all sharing a common configuration, passing through the membrane 7 times. GPCRs are present on all cells in the body. They pass signals from a variety of extracellular messengers to modulate intracellular pathways via the activation of G proteins and other signaling molecules. GPCRs mediate an incredible array of essential actions in the body, including smooth muscle relaxation and contraction, neurotransmission, immune regulation, and overall metabolism, and thus have enormous potential as therapeutic targets for multiple diseases. GPCRs are the largest and single most important family of drug targets in the human body. 25 to 30% of current drugs target GPCRs, including many of the best-selling drugs. Despite this, drug development has been encumbered by a lack of structural and mechanistic information about GPCRs and how compounds interact with them. The predominant problem is that GPCRs are too unstable to easily purify, and they lose their highly organized structure and activity once removed from the cell membrane, precluding them from use in structure-based drug design. To overcome this problem, Heptares developed a structure-based drug discovery (SBDD) platform to transform GPCR drug discovery. Heptari Star stabilized receptor technology forms the backbone of its integrated SBDD platform for targeting GPCRs. STAR technology involves the selection of mutations to the protein sequence, which increase thermostability and stabilize GPCRs in their natural states. Once produced, the STAR GPCR can be purified from cells and still maintain its shape and function. The STAR GPCR can be readily crystallized together with drug hits to determine the detailed shape of the GPCR and ligand interactions, which can reveal new binding sites and opportunities. A variety of different compounds, ligands, can be screened in silico for their ability to fit the receptor. This information facilitates the precise design of drug candidates, with an ideal fit to increase selectivity and a better potential for development success. STARs can also be used to unlock the power of fragment screening for intractable GPCR targets and to help discover novel GPCR targeting antibodies, which can have advantages over oral drugs for some diseases. Through this technology, Heptares is building an exciting pipeline of new medicines to transform treatment options for Alzheimer's disease, schizophrenia, cancer, migraine, metabolic disease, and other indications.
So it's exciting, right? Because a lot of our body and the basic functions which we perform are via GPCRs only. I hope you guys can see my slides. Yeah. So now, having this very brief introduction to GPCRs, now we'll dig into the theory of it and we'll see how they are actually working, correct? And via this we'll understand this part of the cell sorting also. Okay, so what are G protein? So let me get the laser pointer. Wait, yeah. So G protein coupled receptors and second messengers. As their name implies, G protein coupled receptors, that is GPCRs, are receptors that act through a member of the guanosine nucleotide binding protein or G protein. So what is G protein? G protein is basically guanosine nucleotide binding protein. Three essential components define signal transduction through GPCRs. A plasma membrane receptor with 7 transmembrane helical segments, as you have just seen in the video. A G protein that cycles between active, that is when GTP or guanosine triphosphate is bound, and inactive, that is when GTP or guanosine diphosphate is bound, diphosphate and triphosphate. Okay, and an effector enzyme or ion channel in the plasma membrane that is regulated by the activated G protein. An extracellular signal such as a hormone, growth factor or neurotransmitter is the first messenger that activates a receptor from outside the cell. So this is the ligand or the signal ligand, which we have just understood with respect to the signal transduction phenomena. Okay, ligand binding to the receptor forces an allosteric transition that allows the receptor to interact with the G protein, causing it to exchange its bound GDP for a GTP from the cytosol. So GDP bound, that is guanosine diphosphate bound structure, is inactive. GTP bound, that is guanosine triphosphate structure, is active. So what will happen? As soon as the ligand comes and the signal ligand gets bind with the GPCR, it will convert the GDP bound structure to GTP bound structure. Okay, the G protein then gets dissociated from the activated receptor and binds to the nearby effector enzyme, altering its activity. The effector enzyme then causes a change in the cytosolic concentration of a low molecular weight metabolite such as cyclic AMP or inorganic ion, which acts as a second messenger to activate or inhibit one or more downstream targets often protein kinases. Okay. The human genome encodes just over 800 GPCRs, about which 350 for detecting hormones, growth factors, and other endogenous ligands. So what are endogenous ligands? These molecules which are generated within the body, and up to 500 that serves as olfactory, gustatory receptors. The largest superfamily of proteins encoded in the human genome, that is GPCR, have been implicated in many common medical conditions, including allergies, depression, blindness, diabetes, and various cardiovascular defects. GPCR mutations are also found in 20% of all cancers. So you can see, if there are around 800 GPCRs which are encoded by our genome, so chances of them being involved in most of our diseases is pretty high, right? So it is observed also that almost 20% of all cancers which we are aware of, which we know of, they have GPCR mutation associated with them. Okay. In the United States, more than a third of all pharmaceuticals on the market target a GPCR. For example, the adrenergic receptor, which mediates the effect of epinephrine, is the target of the beta blockers prescribed for such diverse conditions as hypertension, cardiac arrhythmia, glaucoma, anxiety, and migraine headache. More than 100 of the GPCRs found in the human genome are still orphan receptors, meaning that their natural ligand is not yet known or identified. So you can see how active area of research this GPCR signaling in, understanding how GPCRs work, understanding in which function they are involved, understanding to which effector enzyme or protein they are associated with. Still very less we know of it. Okay. The adrenergic receptor, with well understood biology and pharmacology, is the prototype of all GPCRs, and our discussion of signal transducing systems begins there. Meaning, if we are talking about a signal transduction, okay, so signal transduction is an important function of membrane protein. Out of these membrane protein, majority of the proteins belong to 7 transmembrane kind of proteins which are called GPCRs. Not all 7 transmembrane helical proteins will be GPCRs, but most of the GPCRs or all of the GPCRs will be 7 transmembrane proteins. Okay. So this is a kind of big video which will actually describe the core functionality and the core understanding of GPCRs. So I guess we can take this in our next session, because GPCRs in itself is a huge topic to discuss. So for me, the challenge was to actually limit my discussion for this particular topic, because a lot of work is being done in this field. Okay, so we're gonna switch. We will try to understand more of it, and I guess I'll try to give you guys more simplistic examples of how GPCRs are working in our everyday life. It will help you and maybe create some curiosity in you guys to go dig more into the research which is being done in the field of GPCRs. Okay, so this is all from my side. Thank you for your time and your patience. Thank you so much.